These are the lecture notes for Chapter 3 on cells. This is a review if you've had biology, but it's definitely information that you're going to need in anatomy and physiology. And I also cover Chapters 2 and 3 because a lot of this information is on the TEAS test. Many of you take that TEAS test to get into your healthcare programs. So this is a good chapter for reviewing for the TEAS test. So the cell is the basic living unit of all organisms. And cells contain, at least human cells, contain organelles, which are, or we sometimes call those cell parts. Sometimes when you're a kid and you study cells in school, you have to make a model of the cell or draw a model, and you'll have to make a model for the nucleus and the mitochondria and the ribosomes, endoplasmic reticulum, Golgi apparatus. There's a lot of different ones, but they're basically little cell organs, organelles. The organelles have all different kinds of jobs that they perform in the cell. The cytoplasm is a jelly-like substance that surrounds the organelles. And then the cell membrane or the plasma membrane. I tend to use plasma membrane, and that's probably because I'm a biology instructor. But the cell is surrounded by the plasma membrane. And this is a picture of a generalized cell, a generalized animal cell. And you can see the different organelles as well as the plasma membrane. You've got the plasma membrane here on the outside, and then you've got a nucleus, and the nucleus is surrounded by the nuclear envelope. Inside of the nucleus is the nucleoplasm. Instead of cytoplasm, it's called nucleoplasm. The DNA in the nucleus is all spread out and diffused. Instead of being condensed into chromosomes, it's in a form we call chromatin. But the chromatin is the DNA. And then inside the nucleus is a structure called the nucleolus. And the nucleus does have holes or pores that allow certain molecules in and out of the nucleus. The nucleolus actually makes, these little dots are called ribosomes. It actually produces the ribosomes. Let's see. Outside the nucleus is the rough endoplasmic reticulum, or rough ER, which is a system of channels that basically makes proteins and modifies them by forming them into complex shapes and different things like that. But the rough ER is outside the nucleus, and then we have the smooth ER, which is more for modifying lipids and carbohydrates rather than proteins. Then we have free ribosomes in the cytoplasm. So anytime you see ribosomes, that means that ribosomes make proteins. Centrioles are these, they almost look like a stack of logs or something, I don't know, but they a bundle of logs or sticks. Anyway, the centrioles are important when the cell divides. And the centrosome is the centrioles and the spindle fibers or microtubules that connect to your chromosomes and pull them apart during cell division. So the centrosome and the centrioles are important in cell division or mitosis. The mitochondria is where glucose is broken down and ATP is produced, ATP energy. The Golgi apparatus takes the products from the ER and modifies them and packages them into vesicles, sends them outside the cell or sends them somewhere inside the cell. Lysosomes digest. Peroxisomes also digest, but they specifically break down fatty acids. So So cells have various functions, which I'm not going to read through, but we are going to go ahead and talk about the cell membrane or the plasma membrane. The plasma membrane is the boundary between the cell, the material inside the cell, and then its outside. So the cell membrane or plasma membrane encloses the cell. It supports the cell contents. It acts as a barrier, a selective barrier, because it determines what materials can come in, what materials can come out, and then also what materials cannot. The plasma membrane plays a role in cell communication with other cells. We describe the plasma membrane as a fluid mosaic model. The fluid mosaic model is the model used to describe the structure of the plasma membrane. And it says that the membrane contains phospholipids and cholesterol, proteins, and carbohydrates. The phospholipids actually arrange themselves into a double layer or a bilayer. And the phospholipids contain two regions, a polar region and a nonpolar region. Basically, a region that is hydrophilic or polar is the head of the phospholipid, and the nonpolar region are the fatty acid tails, and they are hydrophobic. They do not like water. So, So basically the polar region are these little blue circles. The phospholipid looks a little bit like, every phospholipid is like one of these little blue circles with two yellow tails attached to it. So this is your double layer or your bilayer of phospholipids. And the heads of the phospholipids face away. So they either face outside to the extracellular fluid, or they face inside the cell, which is also fluid. So the heads are polar and they are hydrophilic. They are water soluble, water loving. The tails are nonpolar. They are the hydrophobic nonpolar region of the phospholipids. Okay, so the phospholipid has a polar head region that is hydrophilic, and two nonpolar tails that form the tail region, and that is hydrophobic. And that is why you see the tails kind of turned toward one another and away from the watery environment. You also have carbohydrate chains, which can be attached either to these large protein molecules within the bilayer, the phospholipid bilayer, or they can be attached directly to the phospholipids. You see these little, I'll blow this up so you can see it better, but you see these little cholesterol molecules. They have 4 rings, and you see them scattered throughout. The cholesterol molecules help maintain the plasma membrane. They help make it the consistency that it needs to be. So most of the time, cholesterol functions to make the plasma membrane less liquidy and more solid. So that's most of the time what cholesterol does. But if the environment is a really, really hot environment, like in a desert, some organisms, cholesterol actually makes the membrane less fluid and more solid, but some it makes it more fluid and less solid. It just depends on the temperature of the environment. But for us, cholesterol is right here. It adds strength and stability to the plasma membrane. It limits the movement and the fluidity of the phospholipids. The phospholipid bilayer is a double layer, has a fluid quality because the phospholipids are able to move. They don't just sit still, they're able to move, kind of squeeze past each other. There are proteins, large proteins found within the phospholipids that have different functions. And then there's carbohydrates that can be attached to the phospholipids or the proteins. They make the outside of the cell kind of sticky, and they also help identify the cell to your immune system so that your immune system can recognize that this is one of your cells and not attack it, like it would attack something foreign. The plasma membrane is selectively permeable, so it determines which substances can pass across it and which can't. Inside the cell, you're going to have certain proteins, certain enzymes, and certain proteins that are found inside the cell in higher concentrations than outside the cell. And you definitely have a lot of potassium inside the cell. Potassium is found at much higher concentrations inside the cell than it is outside the cell. Now, sodium, calcium, and chloride have higher concentrations outside the cell. So just remember, sodium concentrations are high outside the cell and potassium is high inside the cell. That will be definitely important. Okay, then we talk about transport across the plasma membrane, and that transport can be passive and not require energy. Passive transport does not require the cell to expend energy or to use ATP for energy. Passive transport can just be simple diffusion through the phospholipids. It can be osmosis, which is the diffusion of specifically water. Typically, that happens through these protein channels called aquaporins, but water is small enough that it can go through the phospholipid bilayer just because it is small. And then there's facilitated diffusion, which is passive, but it requires a protein to transport the substance across. So diffusion, osmosis, and facilitated diffusion are passive transport processes. Active transport does require energy. It requires energy, usually in the form of ATP. So it does require energy. Usually that energy is in the form of ATP. So some names for active transport are just active transport, secondary active transport, endocytosis, and exocytosis, which is transport by a vesicle. Bulk transport, large amount of material. When it's endocytosis and exocytosis, it's a very large amount of material. Now, diffusion means that movements are going to move down or with their molecules are going to move down or with their concentration gradient. And when we talk about diffusion across the plasma membrane, it does not require energy. And so basically, molecules are going to move from where their concentration is high to where their concentration is low. That means down or with their concentration gradient, from where their concentration is high to where it is low. And no energy is involved in diffusion through the phospholipid bilayer. So we need to talk a little bit about a solution, and solutions in the human body are aqueous solutions. So the solvent is water. The solvent is what does the dissolving. And then the solutes are the substances dissolved in the solvent. So typically in our body fluids, solutes can be polar covalent molecules like glucose, amino acids, and they can be charged particles called ions. So they can be like sodium ions, potassium ions. But whatever it is dissolved in water, water is the solvent, and the particles that are dissolved in the water are solutes. Okay, so with diffusion in the body, materials that diffuse into a cell or out of a cell actually diffuse through the phospholipid bilayer. They actually cross the phospholipid bilayer. So we call this diffusion when molecules move through the phospholipids. So materials that can cross the plasma membrane directly through those phospholipids include oxygen and carbon dioxide gas. Chloride ion, I'm not sure why, because it does have a charge, so you wouldn't think, but it's listed here. I'm not going to highlight it, but it must be a substance that can cross through the phospholipids. And then steroids, which are also lipids. So if it's lipid soluble, like steroids, oxygen, carbon dioxide, and urea is another substance. But anything that is non-polar, a non-polar substance like lipids, steroids are non-polar. So any steroid hormones like testosterone or estrogen or any other type of steroids are non-polar and insoluble in water, so they can pass through the phospholipid bilayer. Oxygen and carbon dioxide are also nonpolar and they're small, so they can pass through the phospholipid bilayer by diffusion, and they're just going to move from where their concentration is high to where it's low. And then we have osmosis, which is a special type of diffusion. It's a diffusion of water, and water is going to move from where water's concentration is high to where water's concentration is low. And even though water is polar, the reason it can pass directly through the phospholipids is because it is also very small. So water can pass through either the lipid bilayer, because it is so small, or through water channels called aquaporins. So what I always remember is we know which way water is going to move because water follows salt. And we can also say water follows solute. So if salt is your solute, water is going to move where there's more salt. Water is going to follow or move toward the higher solute concentration. So water is always going to follow solute. It will always move toward the higher solute concentration. It will move to where it's more salty or more concentrated with glucose, whatever the solute is. So what we're looking at here is we're looking at something called a U-tube. No, I'm sorry. This is not a U-tube. This is just a tube that's covered with a membrane. I'm sorry. I don't see a picture of a U-tube, so I feel stupid. But anyway, we've got a tube here that's covered with a semi-permeable membrane that's designed to act like the plasma membrane. And what we've got inside of the tube is a 3% salt solution, and outside the tube is just distilled water with no salt. So 0% salt outside, 3% salt inside. Without talking about and making this complicated, if you'll just remember, water moves toward the higher solute concentration. Water follows solute. That will help you so much in A&P because it's going to come up again in the urinary system and other body systems. So the water in the beaker is going to move through that membrane into it's going to follow that solute, that 3% solute. So it's going to move where the higher concentration of solute is. And you can see that it's going to move into that tube, and the tube fills up with water, and it leaves the beaker and moves into the tube. And it's going to dilute that 3% solution. Not to zero, but it's going to dilute it anyway. So osmotic pressure builds up when there is a difference in solute concentration across the membrane, inside and outside the membrane. So if the solution is hypotonic, if a cell is placed in a hypotonic solution, that's like this. If this tube with the 3% salt solution inside, if that's a cell, and it's placed in this beaker of distilled water, that distilled water is hypotonic in comparison to the fluid inside of the cell. If the concentration of solute is the same inside and outside the cell, we say the solution is isotonic. And if the concentration of solute is higher outside the cell, we say it's hypertonic. The solution inside of our tube is hypertonic compared to the solution in the beaker. And the solution in the beaker is hypotonic compared to the solution in the tube. So if a cell is placed in a hypotonic solution, the cell is going to gain water and it's going to swell. Sometimes it will swell and burst, and we'll call that lysis. So in a hypotonic solution, a cell will swell. It will fill with water. In an isotonic solution, a cell will stay the same size, and if a cell is placed in a hypertonic solution, it will shrink. And we call that shrinkage crenation. So red blood cells placed in a hypotonic solution will swell and burst, and we call that cytolysis or hemolysis. I don't know if they've got the word on here. It says it will lyse, but I don't see the word hemolysis. But when that red blood cell bursts because it swells up so big in a hypotonic solution, we call it hemolysis. It just means red blood cell bursting. In isotonic solution, the red blood cell remains the same size. In a hypertonic solution, the red blood cell will shrink, and we call that crenation. And we'll stop here, and we'll start with facilitated diffusion, and that will start our next recording. It will start on slide 26.